Sex determination is actually pretty complicated. For our purposes today, we're only really talking about the chromosomes themselves, not all of the pathways that are involved. So for example, platypi can have up to 12 sex chromosomes, so 2 is not always the norm. There are several types of sex determination that you should be able to define, and a couple that you should be able to use. And we'll get practice using them as we do certain types of genetic questions and problems. But for this section, you just need to understand how sex is determined. So not using them in problems yet. For example, the most common types of sex determination are the standard type that you're aware of, that is XXXY sex determination. But there's also X-based determination, where it is the number of X's that determines what's happening. And there is reversed ZZZW. In this case, the female is the heterogametic sex, or the one that is different. Whereas in the XXXY system, the female is the homogametic sex, the one where they're both the same. There's also genic sex determination, which happens when there are no sex chromosomes. And sometimes things are developed by environment. For our problems, there are going to be a few systems you should be familiar with and that we'll get practice working with later in the semester. The first one is the protenor system. This is the system where there are only X's and the number of X's determines the sex. This means that individuals that receive 2 X's are always going to be female, and individuals that receive 1 X are always going to be male. In ZZZW systems, like in birds, the female is the heterogametic sex and the male is the homogametic sex. Remember that hetero means different and gametic just means gametes. So Z and W are different gametes. So this is heterogametic, whereas Z and Z are homogametic, meaning that they're both the same. Birds work exactly the same as you, but the sexes are opposite. So if you see Z's and W's on a problem in your book or a problem that you might encounter in your practice or exams, you are always going to be referring to birds. And they work this way. And haplodiploidy, whether or not the organism is haploid or diploid, determines its sex. In these cases, females are diploid, males are haploid, and there are no sex chromosomes. And this is really common in ants and bees. So in this case, the only one that's able to go through meiosis is the female. She's diploid. Males are essentially clones or unfertilized eggs, and they are haploid. There are 2 XXXY systems. These you'll have to know pretty well because we will be working with both of them over the course of the semester. One of them is your system, the human system, but the other one is the system for the fruit fly. Fruit flies are one of the most common genetic organisms. So this is a genetic model organism. And while models are helpful for learning a lot of materials, they are not identical to how humans work. While females are XX and males are XY, it is not the presence of the Y that determines if the male is male, but rather the ratio of X's to autosomes. So what do I mean by that? Well, we talked about how in humans, XO individuals are female They have Turner syndrome. And XXY individuals are male and have Klinefelter syndrome. In flies, it is the opposite. XXY individuals are female and they are normal, and XO individuals are male and they are sterile only because there are some genes on the Y needed for sperm development. This is because the Y does not confer maleness. Instead, this ratio is what's important. The ratio is X to A, where X is the sex chromosomes and A is the autosomes. When the ratio is 1 to 2, i.e. XY to a normal diploid, this results in 0.5 being the ratio, and that's the threshold to be male. This means that XO individuals are still 0.5, so they'll develop as normal males. Anything that's less than 0.5 results in a strange intersex metamale that's sterile. The presence of two X's, or a ratio of 1, XX to 2A, results in femaleness. When the value is greater than 1, you also get metafemales, which are sterile. So instead of using the presence or absence of X's, it's the relationship of the number of X's to whether or not the animal is diploid or triploid. If the XA ratio is between these, then you get an individual who is intersex and will develop different traits from both males and females. So to put that into a summary, it looks like this. All of these individuals are diploid, and these individuals are triploid. Anytime the ratio is 0.5, you develop as male. So if you have XY or XO, you'll be a normal male. Anytime the ratio is 1, 2 X's to 2, you'll be female. So XXY is female and XX is female. When there are 3 X's, this increases the number above 1, which results in metafemales. And when you are triploid, this changes the ratio as well. Triploid females with 2 X's become intersex because their ratio is under 1. Individuals above and below become metas. You should understand that this is dosage based. So if you were asked about the gender or biological sex in this case, rather, of an individual, and you were told they are XY, but they're a fly, you would say it's male. If you were told they're XXY and they're human, they would be male as well. But if they're XXY and they're a fly, they would be female. So you should be able to recognize what the sex of the individual would be depending on the type of animal it is. You could be asked about birds, in which case you would see Z's and W's. You could be asked about bees, in which case it's diploid or haploid. You could be asked about flies, which are dosage based, or you could be asked about humans. Humans are part of the Ligaeus model. This is the model where the Y is sufficient for maleness. In this situation, the number of X's don't really matter, nor does the number of Y's. If there is a Y, then you will be male. Most of the information on your chromosomes comes from the X chromosome, and the Y only contributes a small amount. And under normal circumstances, females are XX and will give X's to either of their offspring, while males are the heterogametic sex and can give X or Y. This means that the ratio of offspring stays 1 to 1 because the presence of the Y is sufficient. The reason the Y is sufficient is because it contains a special gene called the SRY locus. The SRY locus is located on the short arm of the Y chromosome, and it encodes testes determining factor, which is essential for maleness. The SRY gene is also sufficient for maleness. This means that when it is expressed away from the Y, the individual will be still male. For example, if you start with an egg with an X sex chromosome, if it's fertilized by a Y sperm, it will be male. If it's fertilized by an X sperm, it will be female. This is your genetic sex. XX is female, XY is male. Gonadal sex is determined by the SRY gene. If the SRY gene is present, then you will develop testicles. If it is not, then you will not. It is possible to have a Y chromosome, but not have an SRY locus, in which case your gonadal sex would be female. The next level of sex determination is androgen production. Androgens are produced primarily by the testicles. If the SRY gene was present and the testicles develop, then androgen production will begin. In normal female development, this doesn't happen. However, in order for androgen production to matter, androgen detection must occur. If androgen detection occurs, then you will develop as a normal male. This is your phenotypic sex. However, if androgen detection does not occur, then you will be phenotypically female. So when we're talking about sex determination from a genetic standpoint, the basis of this is just XX or XY. But gonadal sex and phenotypic sex do not have anything to do with sex chromosomes. With the exception of the presence of the SRY gene and androgen detection, which actually does not occur from the Y chromosome. It's possible to have individuals who are XY genotypically but female phenotypically, and XX genotypically but male phenotypically. This happens when translocations occur or mutations occur on the Y chromosome. Since the SRY gene is sufficient, a translocation from the Y to the X creates an X chromosome that confers maleness. Whereas loss of the SRY gene on the Y chromosome confers femaleness. This determines your gonoidal sex, not your genetic sex. Additional problems happen either when androgens are not produced, androgens are produced by mistake, or androgens cannot be detected. The most common way that this happens is through a mutation called androgen insensitivity. In this case, this is an X-linked trait. The receptors required for androgens are not located on the Y. They're produced from the X. Mutations in these receptors result in a failure to develop male features. For example, these are all genetic males who have developed female features because they have androgen insensitivity. If you saw these individuals on the street, you would assume that they were female, and most of them behave as though they are. There are more than 40 known genetic causes of intersex conditions like this. So even though most of the time in our course, we will discuss male and female as being purely genetic sex, XXXY, this is a very simplified view of how the process actually works. For example, everyone in this picture is not a traditional XX female. All of them are intersex, and you can't necessarily tell just by looking. There are several different places if you're curious about these types of conditions or differences that you could look for more information, but more information will not be necessary for the course. You need to understand that human sex determination largely is caused by the presence of the SRY locus on the Y chromosome. You need to understand that changes in the SRY chromosome location can result in development of gonoidal sex that does not match genetic sex. And you should also understand that phenotypic sex is further removed. Sex determination is very complicated. This makes it very difficult to talk about certain things like what is gender, or how does intersex work, or should we provide things like testosterone during puberty? There isn't a simple answer. So the key today for sex determination is this. You should know each of these types of sex determination theories. The proton or sexual identity sex determination system, where it is number of X's that determines sex. The ZZZW system, which is specific to birds, where the heterogametic sex is female. The haplodiploid system, where diploid individuals are female and haploid are male. The genetics balance theory, where the presence of the Y is not sufficient to determine sex, but the ratio of X's to autosomes is. And the Ligaeus theory, the mode of sex determination for humans, where the SRY on the Y chromosome is sufficient for maleness. You should be able to recognize what sex individuals should be based on the system you're provided and the combination of chromosomes that that individual has. Later, we'll be using these different types of systems to do genetic testing and crosses. For example, if we do a cross in birds, you should remember that the female is the ZW. If we do crosses in Drosophila, you should remember that the number of X's and Y's are not important for determining sex, but it's their ratio to autosomes. Most of the studies that we'll be doing will be in the standard Ligaeus system. So if you're still not comfortable with these other systems for the rest of the course, it's mostly okay.